out. Yeah, so with our live stream, it captures, I think, you and the screen. And it's not like
sure I'm not out.
Yeah, I mean, we can say it's not quite like the quality of a Zoom thing. So if you come in person, you get a better experience than if you watch it through the live stream. Get started. Hello to everyone in the room as well as anyone tuning in through the GSI live stream. I'm Nelson Lau, the director of the Genome Science Institute, hosting on behalf of Ala Grishak, who would normally be hosting our seminar series. It's my pleasure today to be hosting Ignaty Leshchiner from our newly formed Data Science Corps. He comes from the Broad Institute, where he was a group leader working with Gad Getz on cancer genomics. And then we recruited him to BU as an associate professor pending. I guess your associate professor promotion will work through the ranks, and then you'll be part of the computational biomedicine section in Department of Medicine, as well as one of the members of the Data Science Corps to help bring genomics and bioinformatics to other labs that may have issues, getting that kind of expertise on their own. Ignaty is part of this group with also Chao Zhang as well. So we're really glad to have him here. He's an expert in cancer genomics, having carried out a lot of these large-scale analyses of all these different cancer genomes and trying to establish some sort of signature that could be more predictive of cancer diagnosis. He's actually, even before we recruited him, he was already affiliated with Avi Spira's group as a, was that a subcontract or contract?
Co-PI. Yeah,
co-PI with Avi Spira on the Lung PCA, a multidimensional atlas of pulmonary pre-malignancy. I think he'll continue talking about that. Before we hear Ignaty's talk, I just also want to remind everyone that the GSI Research Symposium is going to be held in person this November, and we're looking for abstract submissions by October 14th, so please don't ignore all the messages that Abby will help send out to all of you. And we're kicking off our GSI seminar series with Ignaty as our first speaker, and looking forward to your talk. Thank you.
Thank you. Thank you for the introduction. So, good afternoon, everybody.
Is your microphone on? Let's make sure it's on. Okay, great.
Yeah, thank you for having me here. It's a pleasure really to talk about my work on clonal heterogeneity, competition, and resistance in different stages of the disease. So as we all know, cancer develops along a characteristic path starting from a normal somatic cell.
You might have to hold it up closer.
Oh, it's on, but it's on.
Let me see if it's try that now.
Yeah.
There we go.
Great. Yeah, the cancer starts on a characteristic path where the normal somatic cells start to accumulate mutations. Usually first there are passenger mutations. At some point you can develop a driver that leads to clonal expansions, potentially immune evasion, and eventually to full-blown cancer. After treatment, there's usually a bottleneck, but for many people, unfortunately, resistance develops, and that leads to advanced cancer, metastatic spread, and often death. So we can kind of delineate three major parts in this progression trajectory. Really the pre-cancer age where cancer is not detected yet and it's not malignant. Then when it's already malignant but it's progressing before treatment, basically natural competition of different clones. And then the phase after treatment where it really depends on the treatment resistance and the effect of the drugs, active metastatic spread, and resistance to further therapy. So we developed tools really to analyze all stages of this disease, and I'm going to structure my talk actually in three parts, really delving on all of these three areas. And so we have tools really to model progression of cancer before diagnosis, which tends to model order of somatic events and the way the cancer develops. Then really modeling the growth kinetics and clonal structure of clones during natural progression. And then really try to delve into the metastatic spread and phylogeny of different clones spreading in metastatic disease, and understanding, for example, through autopsy analysis. As you can see, some of these tools were used in multiple publications quite successfully, so some of the publications here I'll actually talk about in detail. So again, my talk is going to be about three parts. So on the pre-cancer progression part, I'll be talking about pan-cancer analysis and progression trajectories across multiple cancer types. For the natural progression phase, I'll be talking about a study we did in leukemia, looking at clone competition and driver fitness defects on natural progression. And the last part would be some more recent work, actually studying how cancer spreads through the body, through analyzing autopsies and cell-free DNA from these advanced patients. First, I'll briefly go over the methods with some brief details. The first set of methods is, we call them timing. It's a little bit of a non-intuitive approach where you can look back into the history of the cancer by analyzing the sequencing data at time of diagnosis. So the way it works is that when cancer grows and cancer cells mutate, any mutations that were accumulated on any of the chromosomes get co-amplified with the chromosomal gains. So as you can see, the red mutations here are now doubled up and they're present in both copies. That allows you to record effectively mutational time when these copy number events happen. And since copy number events happen along the development of the cancer, you can actually figure out the trajectory and the timing of all these individual events related to those copy number events. So based on the copy number events, you can also define timing of individual mutations relative to that copy number gain. You can define other somatic copy number alterations like small deletions and other small gains relative to it. You can also time pretty much any event across the genome by the use of whole genome doubling, which is a genome-wide event that allows you to really put time on multiple different chromosomes together. And just for example of the raw data here, these are allele fractions of somatic mutations in whole genome, so ovarian and esophageal cancer here. So we're looking at an area of chromosomal gain, so 2 plus 2 means there's 4 copies to each allele, and this is a 2 plus 1, so just one gain of one of the chromosomes. And we can see these are lines that are corresponding to what we call multiplicities, basically, if the copy number of the mutation is 2 or 1 in this situation. So if the mutations happen before the gain, there will be a lot of multiplicity 2 mutations, meaning you're going to have large peaks in this area, like you see in this cancer here, meaning this gain actually happened quite late after a lot of mutations had been accumulated. In case of ovarian cancer here, we see the mutation actually happened quite early. There's not a lot of mutations accumulated before the gain. Most of the mutations had been accumulated after the gain. So, these are little fraction distributions we can now explore in a more probabilistic model when actually each area of the genome is modeled for probability of belonging to multiplicity 2 or multiplicity 1 mutation, depending on the copy number stage. We need to correct for detection, power issues, coverage, and really transform this probability of being a multiplicity 1 and multiplicity 2 mutation into some sort of a timing estimate, which we call pi, which is defined from 0 to 1, meaning 1 is the somatic event had been developed at the latest point during the diagnosis, and 0 meaning germline cell, basically no mutations had been accumulated. Here's an example of one of the genome, which actually happened to have a whole genome doubling, and there's different copy number regions, 2 plus 2, 2 plus 1, 2 plus 0. We can see that all of those gains here, since it's a genome-wide event, actually happened roughly at the same time, but there are some gains that are happening later, and they happened about 80% of this mutational time pi. So the next phase is how to model these clonal populations, different subclonal populations in the cancer. So we developed a method that is n-dimensional, meaning that it can handle multiple samples at the same time and jointly infer the clonal structure of those tumors. The way it works, it's a multidimensional Dirichlet process. Basically, clusters cancer cell fractions of individual mutations, and this n-dimensional space. Here we have slices of 2-dimensional spaces and this n-dimensional space, and you can see that different clusters separate well in this space. And here we have 4 samples basically where we see how different clonal clusters move throughout time, for example, in this patient. The next phase, after you cluster the mutations, you actually need to define the phylogeny of those clusters. Why? Because you need to assemble genotypes of individual cell populations, so effectively you need to build a phylogeny. There's multiple different phylogenies, especially if you have a large number of samples and a large number of clones. Like here we have 12 different clusters. So the way we do it, we do a Monte Carlo Markov chain process based on a probabilistic pigeonhole principle, which basically means that any parent of a clone needs to have a higher CCF than its children. And the way it's sampling here, as you can see in this short video here, basically the clusters are moved around the tree, and the probability is sampled in the Markov chain process. And we arrive basically to the composition of samples, where each sample now is composed of different cell populations. We use these concentric pie charts to represent the phylogeny and the composition at the same time. So basically, this purple clone also has the orange and the green mutations in the cells. So now to some interesting results we get through these methods. So the first part is going to be talking about this early stage of cancer development. So we can apply these methods, and basically on a patient by patient scale, we can resolve the trajectory of the cancer development. And we can see here driver events being ordered along these timelines. So most of these events actually happen before the diagnosis, or they're all clonal, but you can still order them. We see here different subtypes of lung adenocarcinoma. There's these fusion-driven cancers that we actually see some splice factor mutations here early on. For EGFR and KRAS-driven cancers, we see that the main driver mutation is actually very early, followed by corresponding gains on those chromosomes. And for p53-driven cancers, we see p53 as an early mutation and followed by whole genome doubling. So if we take a large cohort of these tumors and use probabilistic method to assemble some sort of an average trajectory for this cancer subtype, we arrive in this probabilistic, we call it the league model, basically probabilistic ordering of events for an average cancer in this subtype. Remarkably, if you find earlier work where experimentally people were able to obtain premalignant tissue and sequence it at different stages of disease, like this well-known work in colorectal adenocarcinoma, the order that we can reconstruct for primary tumors computationally matches very well with the well-known experimentally validated order. You can see ABC followed by KRAS and p53 and etc. Events ordered correctly here. This is another example in head and neck cancer, which again for HPV negative cancer, we're able to obtain early stage premalignant lesions at different stage of progression. And again, the order matches very well with the copy number events that were detected in this early study in the 90s. Obviously, for most cancers, it's actually very hard to obtain premalignant tissue, especially at different stages of progression. So we can actually apply these methods to now understand progression of cancers where we can't really obtain premalignant disease. And head and neck squamous cancer is actually a very good example because there's two major subtypes of head and neck squamous carcinoma is HPV negative and HPV positive. So basically a viral driven form of the cancer and the smoking driven form of the cancer. So for the HPV negative, as I showed just before that, we have tissue that can be obtained from the mouth for different stages of disease, and it progresses here. For HPV positive cancer, it usually progresses into the tonsils, and it's impossible really to capture that stage when it's premalignant because it's not seen and it's not detectable. By the time it's detectable, it usually already spreads to the lymph nodes. And interestingly, the order of progression is quite different for HPV negative positive. There are some commonalities, but the most remarkable things is that tumor suppressors are being deactivated here by copy number losses, and mutations like p53, CDKN2A, etc., are actually being deactivated here by the virus, and we don't see these events as early events. So we apply these tools across large number of cancers in this big pan-cancer analysis of whole genome, along with other groups. We're able to really reconstruct the trajectory, the average trajectory for main cancer subtypes, defining the major early events, events that happen later in progression, and late or subclonal events that occur after diagnosis and potentially with start of treatment. And so here are some examples here, here, for example, for non-Hodgkin's lymphoma, we actually see immune-related genes being early, like B2M mutations that deactivate HLA presentation, being very early mutations. For clear cell renal carcinoma, we know that the HL is the main driver of this cancer, and we actually see both the loss of chromosome 3 and VHL mutations as early as been known for this subtype. So just to summarize this section, what we can do next, really with these methods, is we can understand or try to understand how the premalignancy actually transforms to cancer. And for example, this project Nelson mentioned for the human tumor atlas network, we have pre-cancer projects there that actually try to understand how the progression happens. Are there any sort of dead-end trajectories? Why some lesions that seem to be on the path to progress actually never progress to cancer? It's also important for early detection and prevention, because you can envision the order of events and understand if these events that you're detecting are worrisome or they're really on a dead-end trajectory and you should not be worried about it. Next is really important that you can also do this ordering, not just on the pre-treatment samples, but actually post-treatment samples. And when you only have a post-treatment sample, you can actually try to understand what was the progression and what are the main drivers of that later stage progression, and try to target these important drivers that led to progression or clonal. The next stage is really to be able to reconstruct in more detailed way the progression of each patient, and really maybe model the treatment or the choice of treatment for these patients based on their individualized progression trajectory. So next I will actually talk about this natural progression of cancer before treatment. We had this work on leukemia, where first we noticed that leukemia as a cancer here develops on two distinct paths of growth as a whole. So some tumors take very long to progress. CLL could be a relatively indolent disease for many years. The patients don't even need treatment. And what we found, some of these patients actually follow a logistic growth pattern, meaning their tumor effectively reaches some size, and then their white blood cell count is not increasing for a long time. Others really progress very fast, and their tumor really grows quickly. Their white blood cell counts just shoot into the sky, meaning that they need treatment urgently, and they're not, for example, responding to treatment if that happens. So we had a cohort of 21 patients here for multiple sampling on pre-treatment timelines, and we are trying to understand what this behavior entails in terms of clonal structure. So we're able to reconstruct the clonal structure with phylogenetic Ignaty for all these patients, and then model the growth rates of individual clones, because we have a very accurate sampling of white blood cell counts over time in these patients. So we were able to identify clones that are really growing faster than its parent or other sibling clones. In this patient, for example, and there's a second hit on the p53 gene, and this orange clone is growing, but also its child is growing even faster because it has full deactivation of p53. It's the most fast growing clone that's really outcompeting every other clone in this patient. So we did it for all patients. So some patients still exhibit this logistic behavior. There's no clones that are growing faster. It's really stable clonal structure for many years. Others really develop important drivers, like we just saw the p53. There's the ATM second hit in this patient. Also, this black clone is really growing faster than anything else. We have a KRAS as a driver that is also really taking off and growing faster than any other clones. Next, we were trying to establish if the logistic growth patterns or the exponential growth patterns of the whole tumor are really dependent on how clones are performing. Are the individual clones growing exponentially or logistically, or are they dying and something else is happening? So we were able to identify clones that are actually growing exponentially, for example, even in a logistic patient. So in this patient, this orange clone is continuing to expand, even though the overall tumor burden stays the same, meaning that at some point soon in the future, this patient probably will need treatment because this orange clone is going to take over the population and then continue growing. In some of the exponential patients, there are clearly exponentially growing clones that are responsible effectively for this exponential growth. But then there's other clones that are either actually growing logistically or slowly, or even dying in the process. So it's really a clone competition here. So where we can apply these methods and these approaches in the future is really to try to understand these growth kinetics of individual clones, how this fitness changes under the drug effect. Can we choose a drug based on the slope, effectively, of the fitness of individual clones, and choose drugs that kill all clones, including faster growing ones. We can apply these methods also in solid tumors by using cell-free DNA to also understand response, growth and death rates of individual clones during treatments, and really understand also the transcriptional profiles of these clones. What is causing these clones to grow much faster than other clones, and if it's something that could be targeted. So the last stage that I'm going to talk about is really the evolution of cancer and this advanced disease. So the first study I want to talk about is a study we did in different GI cancers where we looked at cell-free DNA and paired solid biopsies from multiple patients. This is a study done together with Ryan Corkin and Apara Parikh at MGH. So when we did analysis of regular solid tumor biopsies for these resistant patients, we really couldn't identify resistant mechanisms for over half of those patients. And most of the patients show just one potential alteration that could be responsible for resistance. When we compare that to cell-free DNA from the same patients, the proportions really changed. First of all, for the majority of the patients, we were able to identify at least one resistant alteration. And then remarkably, for many patients, there were actually more than one alteration, including some patients have multi-site resistance and having 8 or 5 different mutations in different clones, effectively, that are resistant to the same drug. This is very important clinically because when you try to treat the patient, you really need to attack all the different clones and all the different alterations. If resistance develops in such an easy progression where there's multiple different drivers being hit, it's really something to think about how we approach treatment. This is a patient where we had cell-free DNA, but also we had a post-this autopsy, and we're able to really analyze how the cell-free DNA correlates with the spread of the disease across the body. So this was an FGFR2 resistant being treated patient that became resistant. And remarkably, the original tumor that was in the stomach have not even shown an FGFR2 fusion. So the green clone here that's in the stomach actually has a 53 mutation, but it doesn't have the FGFR2 fusion. So that means that it will probably not even respond to the drug itself. The progressive disease that spread did have the fusion, spread to the liver and other parts of the body. But each of these clones actually developed their individual resistant mutations in FGFR2. So we can see the 617 V mutation in these orange clones, and then we also have the V clone that has another FGFR2 mutation. If you do a solid biopsy of each of these individual lesions, they're pretty homogeneous. Some of them are quite homogeneous in their clonal structure, so all these mutations will appear clonal. But when you do cell-free DNA, you actually see a sampling of all of these different lesions. So their DNA is shed from multiple locations in the body, and it's really kind of a soup of different clonal structures that are being captured by cell-free DNA. And actually, you see that post-biopsy, the resistant clones kind of take over most of the cell populations. So basically, that means that we can try to search for these multiple resistant mechanisms for a specific drug and try to cover for them in the future. So in one patient, effectively, there's multiple subtumors that have different resistant mechanism. And if you analyze the clonal structure of those tumors and you see that there is a clone in one of the samples that is resistant, for example, with a known mechanism like an ACR1 mutation here, and there's another clone that is also outgrowing, but you cannot really find a genetic alteration that could explain, or a known genetic alteration that could explain resistance, it means there's actually a gene, a novel gene to discover, or there may be other epigenetic or copy number mechanisms that are responsible for this clone to actually be resistant and also grow out. So we try to take this further in this study with MGH Rapid Autopsy Program and Dan Urich, where we collected multiple cases from patients that passed away from different tumor types. Today I'll be talking about the breast cancer project that I've been doing. So here we have a cohort of multiple autopsies from samples taken from different parts of the body after death, and also in some of the patients we have cell-free DNA along the treatment timeline or at the end of treatment, and also we have paired biopsies pre and post treatment. So this is a CDK4/6 inhibitor treated ER positive metastatic breast cancer cohort. So first, when we reconstructed these phylogenies and the colon structure of these patients, we noticed that there's multiple patients that have individual sibling clones with different ESR1 alleles. So basically, they're independently resistant cell populations that developed independent resistant mechanism in the same gene, effectively converging on the same pathway to become resistant. And ESR1 by now is a known resistant mechanism for CDK4/6 AI paired therapy. So that was interesting, and that was shown in multiple patients. And these patients are very complicated spread of the disease. We have some clones that are ESR1 mutated, like the clone here and the clone here. And one of these biopsies lesions we actually followed longitudinally during time. So originally the lesion didn't have the ESR1 mutation. After the first treatment, it developed it. And then we have cell-free DNA, which shows that the proportion of that lesion in the cell-free DNA grows over time. So it becomes really an outgrowing resistant lesion. And at the time of autopsy, this lesion was really dominated by this resistant clone. But then there's other lesions that are actually not showing ESR1 mutations. They're showing other drivers. Some of them are unconclusive. We really don't know if they confer resistance. But just 2 of those cell populations are based on ESR1. Others are based on other driving mutations. If we look at just ESR1 across pre-post biopsies, it's clearly contributing to resistance, because in multiple post-treatment samples we see a higher cancer cell fraction of those mutations across the board. But there's also some tumors that have these mutations clonal originally, so they pre-existed before treatment. Potentially those are the ones that are less likely to respond to these inhibitors. But then there's also situations when the ESR1 mutation is replaced with a different one, meaning potentially there's a sampling bias. The biopsy potentially was taken from a different locations in the lesion, and there's actually multiple independently developing risk mechanisms. So to search for new drivers that potentially have a similar mechanism of resistance, basically converging across multiple clones onto the same gene, suggesting that it's an important gene for resistance. We did reconstruct the trees and the phylogeny for all of these patients, and then we performed regular patient gene enrichment analysis and mutation significance analysis, which arrived at well-known breast cancer resistant genes, really not identifying anything novel that would stand out as a resistant mechanism to CDK4/6 inhibitors. If we do this analysis now by clone and also add the aspect of clones converging in the same patients, so they're more likely to be found in the same patient than across the cohort, we start to get novel genes that show convergence across clones and across patients. There's the ESR1 that we just looked at, but there's this new gene that is a KMT2C, which is a known breast cancer driver, but we really see it occurring in a convergent pattern across multiple novel lesions that are growing after treatment. So when we look at some of these patients, we see that sometimes it's paired with a ESR1 mutation, but there's a separate outgrowing population, like on this side of the body, with different KMT2C allele. And we see that further. In this patient, they actually have multiple KMT2C mutations, and they're all at different branches of the tree, suggesting it's really an important gene for the cancer to progress and spread. So KMT2C is a methyltransferase chromatin modifier gene, a very long one. So we first tried to understand what could be the potential effects of the mutations, loss of function mutations in this gene. So when we analyzed RNA-seq from these samples, separating them for KMT2C mutated versus not mutated samples, we identified that the ESR1 is one of the regulated genes in the KMT2C mutated lesions, and you can see otherwise other genes between clonally similar samples are really expression is very stable. When from the same patient you compare clonally different samples, expression actually changes somewhat in specific genes, and specifically in ESR1 here is that it is expressed much less. So that led us to the idea that potentially KMT2C is making these cells less ER dependent. That's why they become resistant to fulvestrant and CDK4/6 treatment combination. So we followed up that with experimental work with the help of Lisa and Thomas at the Broad, and here we're created a KMT2C knockout in a breast cancer cell line that was originally sensitive to fulvestrant and palbociclib, which is a CDK4/6 inhibitor. By introducing indels in KMT2C, we actually make these cells quite resistant. Specifically, they're more tolerant to the drug, but also after 20 days they actually start to grow out much actively compared to the wild type controls. So effectively they change their phenotype to even more resistant, potentially, since again this might need some sort of chromatin remodeling. It takes time for these cells to adapt and really start growing quite quickly. So really the last stage here for this project is really to understand how these individual mutations in individual clones affect expression patterns and which programs they activate during progression. So for that, we actually did single nuclei sequencing for these autopsy patients. And we have both tumor and normal cells here. And then we can use the bulk data with the reconstructed phylogeny for these multiple samples. And we develop methods now to actually map the clonal structure of these individual bulk clones that we define in this multi-sample cohort back to single cell data. And we basically see that different clones show distinct expression patterns. Here in UMAP, we actually see that they separate and they're different locations. And now we can actually do differential expression between different branches of the tree and really say, okay, we have a KMT2C mutation. How does that impact the expression program of these individual cells? And really try to dig deep into how these tumors develop resistance. So to summarize this section, it's clearly that tumors develop multiple resistant mechanisms across the board, especially if they're advanced and metastatically spread tumors. So we can use convergence of these resistant mechanisms, and they can be genomic convergence, they can be converging on transcriptomic or proteomic level, to identify important programs for resistance and try to design, for example, drug combinations to attack these most common resistant mechanisms. It does allow us to actually find resistant mechanisms on much smaller cohorts of patients. Yes, we might need cell-free DNA or multiple tumor samples from the same patient, but we don't have to collect hundreds of patients to identify the most common resistant mechanisms. And we can also try to find even more convergence, sequencing deeper and reconstructing more smaller clones in the tumor and trying to understand if there's other resistant mechanisms that could be contributed. It's also self-controlled because the genomics of the trunk of the clonal tumor is the same for these clones, so you can really focus on just these small changes in expression of small genotype changes that are conferring resistance with keeping the other multiple other clonal events the same. So with this, I'll open it to questions. Definitely many thanks to all our collaborators from Dana-Farber and MGH, the Pan-Cancer Analysis Consortium, and my former mentor's lab, Gad Getz, and all the folks that worked with me on these projects.
Thank you, Ignaty, for that great talk. Questions? We'll start with you.
Yeah, thank you for the talk. I have a question about the first part when you were doing your pre-malignancy analyses. Can you pinpoint exactly how long ago the mutations happened, or do you get more information about just the order of the mutations?
Yes, so to answer your question, so in this study we actually tried to do both. So the methods I showed here estimate this progression in mutational time. So if we go back here, so this is measured in mutational time. So this pi space is basically total number of mutations accumulated in the genome, normalized to DNA amount. So that doesn't give you a wall clock time. It just gives you a progression, and potentially in some situations the signatures could accelerate. So the mutational accumulation accelerates. So that accelerates the time when the tumor develops. But there's methods, and we did this in this study actually. Here you can use aging signatures, signature 1 in particular, that is known to be well correlated with wall clock time for majority of the patients. It is tissue specific, so different tissues accumulate that signature at different rate. But if you have a normal tissue or a tissue that you can standardize that rate across the patients, you can put an estimate on the wall clock time for these events. So we can actually estimate age when the disease happens. And if you can see here, there's actually underneath there's years, which estimates the last clonal expansion, and also roughly the age when different events occur. It's obviously different for different patients. It depends on the patient's age. It depends how long tumor had been developing. But in general, for many solid tumors, we actually see that the tumor had been developing for decades before it becomes diagnosed. It could have been pre-malignant. It could have been a pre-malignant lesion, but it started 20, 30, sometimes years before, with some of the even big copy number events happening early.
I have a question about your phylogenetic tree analysis of these different clones. How do you test the phylogeny tree here? Like, what kind of bootstrapping, or do you use anything as an outroot to root these trees? Or is there what kind of validation? Do you do an experimental validation afterwards?
So you can do experimental validation with single cell sequencing, probably mostly single DNA sequencing. But they also validate themselves. You can basically take away some of the samples and do the reconstruction, let's say just on the 2 samples, and see if that's good enough for the resolution. And then by adding additional samples, you really see the mutations separate. As you can see here, the clones really separate well in this cancer cell fraction space, which is effectively the proportion of cells that the mutation is present. And the idea is that each of the genetic clones, as they grow, all the mutations grow with them. So there's quite a bit of separation in this space between those clones, and the dimensional resolution really helps, because it might be hard to separate subclones in just one sample, but as you can see here over time, these clones really change in their abundance, and that helps really to resolve the structure very well. If you were just analyzing these 2 samples, you probably won't be able to resolve this black clone, because it's And we also extensively generate simulation data where we test that these things work through computational simulations of these tumors.
I had a question for your talk. I'm working on mechanisms of innate resistance with primary patient tumors. I'm not able to, I haven't been able to collect samples over time. Are you still able to use this method to separate potential innate versus acquired mutations from an excess solid tumor?
Yeah, so timing allows you to do that. So one of the applications is not to look into the history of the early developing cancer, but actually look into the history of resistance being generated. So if you have only post-treatment samples, or we have autopsy samples where we don't have any earlier time points, so you can actually reconstruct both of the progression and the order of these events as they lead to progression. So you can delineate events that are late in the progression that are likely to be responsible for the resistance.
I have another question about the comparison between the tumor biopsy and the cell-free DNA analysis. Did I ever correct the cell-free DNA had more clones than the tumor biopsy?
Yeah, so this limitation I think was known for a while, that when you biopsy with a needle, you're just biopsying one lesion, and clearly in metastatic patients, many of them have multiple lesions that are effectively growing independently. And when you treat those lesions, they actually independently develop resistant mechanisms.
So the cell-free DNA is profiling potentially many other potential tumors not identified?
So our understanding is that cell-free DNA is shed pretty much from all locations in some proportion. Liver is obviously a bigger shedder. It might be harder to get brain lesions and bone lesions to really shed much into the bloodstream, but other locations really shed a lot of DNA. It's just the relative proportions, and that really allows you to sample, as we showed in this patient, pretty much all of these clones with just one blood draw. But you do need to develop methods that allow you to do this resolution, because now you have multiple, 10 or 12 cell populations in just one sample, which is not typical for a solid tumor.
Now, how do you reconcile if it's cell-free? Do you worry about just general fragmentation and lower quality of DNA if it's cell-free, or?
Well, the cell-free DNA, there's normal part of the cell-free DNA that everybody has, which from blood cells and other tissues. But remarkably, for many, especially advanced cancer types, a large proportion of the circulating cell-free DNA in the blood is actually from cancer. So in these patients, it's usually anywhere higher than 25% of total cell-free DNA is actually circulating tumor DNA. Which is a reasonably high purity even for a solid biopsy. So purity-wise, for advanced patients, it's actually a very good media to collect. For early stage patients, your abundance of cell-free DNA tumors, cell-free DNA really falls to 1%, maybe 5%. And you really need to develop methods that allow you to sequence deep and understand that structure. But for advanced patients, it's a very good media to really understand the progression. In terms of fragmentation, it's usually nucleosome-bound DNA, so it's about 160 base pairs fragments. Sometimes it's doubled fragments, but so they're shorter reads. So you cannot do any long read sequencing or anything like that, but you can definitely do regular exomes and whole genomes as you would do.
So it's the whole genome coverage, repeats, and everything.
Yes, more or less, it's the whole genome. There are regions that are less covered because of nucleosome positioning, and it's also tissue specific. So different cancers and normal cells, they have different coverages along the genome because of this nucleosome positioning and survival of DNA in the blood. So people are now doing chromatin structure reconstruction for these fragmentation patterns, because you can see bound and non-bound DNA effectively at the coverage. So you need to normalize, if you're doing copy number analysis, you need to normalize these effects, but they're pretty stable tissue specific. So if you know these profiles, nucleosome profiles, you could use that as a normalization.
Okay.
So
Yes, so you can do that. I mean, with these effects, it's just very hard because it's very small number of samples, and you really need big statistics. So you can expand it further and try to do so. There's work that tries to look at negative selection and things like that, but you really need big numbers. So that's tougher than, because here these clones are actually selected for. So you would have never seen them if they were not being drivers. So they're very quickly in a year they're selected for, and they really reach detectable population size. But negative selection, you don't see that. So it's tough.
For malignant disease, how do you differentiate non-malignant clonal proliferation, and what's the event that you separate malignant proliferation from that?
Yeah, so that's a big question, and that's why I think we're doing these studies for pre-cancer, really trying to understand where is the separation in this line of development where it becomes cancer. So clearly most of these events actually develop at the stage when they're not malignant yet. So they're developing in pre-malignant lesions. As we show now, there's clonal expansions in all normal people and all normal tissues. There's always cells that are exhibiting some sort of clonal expansions, especially with age, but they don't lead to cancer necessarily. So what actually triggers this event that causes this to become malignant is still an open question. In some cancers, people think it's like whole genome doubling, for example. When the cell becomes tetraploid, that really leads to progression because cancer now can really alter its proportions of genes easily. There's immune evasion is an important for setting for some cancers, but it's an open question that's people are trying to answer what actually leads to that last straw that caused progression.
Any other questions? If not, let's thank Ignaty for a great seminar. Thank you.